So now we move to the final group of our fellows' presentations, and to start off today, I'm delighted to introduce Professor Prof Varshney. Rajiv, you've been instrumental in developing and applying large-scale genomic resources to crop management. You've determined the genomes of so many species, and it's made a huge difference to broadacre and horticultural crop industries in Australia and around the world. Farmers and people are now eating and thriving in Africa and other places because of your work. Congratulations. I know we want to hear more about it.
Good morning, and thank you very much, Lynn, for nice introduction. First of all, I would like to thank Academy for inducting me its fellow. It's really an honor and privilege for me. And now I would like to deliver my talk. So before I present our research work, I would like to mention briefly that from where I have come. I did my BSc and Master's degree from AMU India, then PhD from CCS University Meerut. Then I had a stint for about 5 years in IPK. This was the time I started to have the collaboration here in Australia, because one of my postdoc project was in collaboration with Professor Peter Langridge from University of Adelaide, and of course, Professor Andreas Graner from Germany. Then I moved to India, where I served ICRISAT, Global Research Institute, in various science and research leadership positions for about 17 years, and continued to have the collaboration with Australia. Then moved to Murdoch University in 2022. During these last 25 years, I had a privilege to work with very eminent scientists, colleagues, and very hardworking people who were the part of my teams as colleagues and collaborators. So I'm thankful to each and everyone, and also had an incredible mentorship from large number of individuals during last 25 years or so. But very quickly would like to mention some of key colleagues: Professor P.K. Gupta, my PhD supervisor; Professor Andreas Graner, my postdoc supervisor; Professor Wajahat Hussain, one of the great teacher in BSc, MSc; Dr. William Dart, Dr. Peter Carvery, Dr. David Worthington from ICRISAT, and Professor Peter Davis, Professor Eastwood, and Professor Andrew Diggs from Murdoch University, and of course my family. And would like to also thank Professor Jan Small and Professor Steve Poblis, who are also sitting in this room, for nominating me for this fellowship. Now, as I'm an agricultural scientist, and we know that agriculture and food is facing lots of global challenges, and some of them, as we know, that overpopulation, ongoing conflicts and geopolitical issues, these makes agriculture much more important in the current scenario from the perspective of the food supply, and of course climate change. All these issues, they pose a serious threat for the food security in developing countries. The good news is that we here in Australia, we play a very key role to contribute towards food and nutrition security around the world. Though in agriculture in Australia, we are having this thing on that on the belt, we call it agriculture belt. In this side, so our arable land, we are having around 25 million hectares, but agriculture worth is about $44.5 billion. So what agricultural scientists in Australia are doing, they need to keep the Australian agriculture climate change ready and internationally competitive. So what we have been doing, when I say agriculture, not only the geneticists or breeders, but all the entire agriculture community, including agronomists, pathologists, etc. So what we have been doing, that we are working towards enhancing yield, quality, and production area. And if we are able to do anything increasement, either in yield, quality of production, we can always enhance our value of agriculture production, can benefit growers in Australia, and even just 5% in any direction can give that advantage of about $2.25 billion or so. Now, here I would like to take you back in 1960s, when many countries in Asia, they were struggling to provide enough food to their population. At that time point, Dr. Norman Borlaug, Dr. M.S. Swaminathan, Dr. Gurdev Ghosh, and Dr. Yong Longping, these gentlemen, they provided high-yielding wheat and rice varieties, which saved many millions of the peoples in Asia through the transformative agriculture by contributing genetics and breeding. And I had a privilege to meet Professor Norman Borlaug in 2003 in Italy, where he challenged the next generation of scientists to embrace modern gene technology to create innovative and sustainable solutions. And these words, they really impressed me. At that time, I was working in Germany. When I went back to lab, and at that time, genomics was in its infancy. In 2005, we had the first crop genome in the form of rice sequenced. This was the same year when I worked with my mentors, Professor Andreas Graner and Professor Mark Sorens, and we provided a framework of genomics-assisted breeding. And then, what we believe that if we can integrate genomics in the crop improvement, we can really deliver the superior varieties, which can be disease-resistant, high-yielding, etc. But to do this thing, we need to understand the genetics of those different agronomic traits. With this objective, what we have been doing during last 15 to 20 years, my lab has been contributing leading sequencing of genomes of large number of crops in collaboration with different colleagues and collaborators around the world, including here in Australia when I was working at ICRISAT, but even now at Murdoch University, we are having genome sequencing center kind of thing, and we are contributing a lot in this direction. So after that, and then we have been using the genomic resources to understand the genetics of agronomic traits and development of the better varieties. I cannot provide everything in this short presentation, but would like to give an example of a chickpea crop, which is a very important crop from India perspective, Australia, but drought is a serious production constraint. So what we did that in 2013, we developed the chickpea genome. Then in 2016, we sequenced around 100 varieties. Then in 2019, we sequenced the germplasm accessions, so like landraces, the genetic diversity, which has not been used in the breeding program. And then eventually, we also decoded genomes of more than 3,000 chickpea lines. We presented this genetic variation map. And recently, we have completed the genome sequencing of about 10,000 chickpea lines. So what we have done that based on these sequencing efforts and phenotyping in the field conditions, we found a QTL hotspot in chromosome number 4. We have dissected it further. But when I was working at ICRISAT, we wanted to integrate this thing in the chickpea varieties. At that time point, we realized two serious constraints in the breeding program. One, cost on the genotyping. So we reduced it by establishing a low cost and high throughput genotyping platform, which is being used right now in more than 28 countries, and breeders can make this, they can make the best use of this platform. Second was the skill set in the breeding program in Africa and Asia. So what we did, we organized different training courses, at least 10 training courses. Through these training courses, we trained breeders in modern genomics and breeding skills. And then we have used the different kind of molecular breeding approaches in that crop improvement program, and our breeders, they developed improved lines based on those genomics integration. And then after rigorous testing in the different locations in the respective countries, several climate resilient high yielding nutritious variety have been released, delivered to smallholder farmers. For instance, in this slide, you can see 5 varieties of chickpea, which is having 15 to 20% higher yield in the case of India, and also in Africa, like in countries like Ethiopia, Kenya, Tanzania, etc. And not only for the drought, we also deliver the varieties for several diseases, Fusarium, Ascochyta blight, etc. Now, when I moved to Australia, we thought, can we replicate similar kind of storage here in Australia? So what we did, we started working with the GRDC, and we developed a chickpea pen genome for Australian varieties. And then what we did, that we did this pen genome analysis here in Australia, and we found that Australian chickpea varieties, they are missing that QTL hotspot, which was responsible for higher yield. So what we did, that together with GRDC, Chickpea Breeding Australia, we started to introgress this QTL hotspot, and we have developed the improved lines, and they will be evaluated next year here in the different locations. Wheat is very important crop in Australia, and what we are doing right now in wheat, that I think last year or so, we have developed a pen genome of 17 wheat varieties, and we try to understand those regions which are responsible for agronomic traits. And right now, we are leading an initiative, and this initiative will deliver wheat pen genome for 100 wheat varieties, and also the haplotype catalogs for 1,000 wheat lines, and many of them, they will be included from Australia, and they will going to change that breeding pattern, what we are doing here in Australia. And what we are doing, that we are using these resources to enhance nitrogen use efficiency, which is a very important trait here in Australia, as well as the heat tolerance in collaboration with large number of partners. Similar kind of efforts we are doing in the horticultural crops, which is also very important industry here in Australia. And what we are doing that together with Hot Innovation and with several partners here in eastern states, we are developing the genomic resources, low-cost genotyping platform in large number of crops. And also recently, we also initiated a project together with Queensland University of Technology in collaboration with Hot Innovation, where we are using the genetics approaches, AI and machine learning approaches to create next generation orchard. So in summary, genomics technology has been very powerful. We have been making the best use of these technologies to develop the new generation of breeding lines. In my opinion, national and international collaboration and creation of next generation scientists is really required for sustainable agriculture. And at this stage, I would like to thank large number of collaborators, partners from Australia, as well as from the international organization. And finally, my sincere thanks to hardworking people from my groups, PhD student, postdoc, etc. Thank you very much for your attention. I will be happy.
Thank you so much. This is just global scale research, and it's great to see it happening here in Australia. And I know there are going to be lots of interest in this, and I know Steve Powell's is in the audience amongst others, so I'm looking for hands going up. And while you're thinking, I want to know more about your training program, please, because one of the great things about agricultural research is how quickly you translate it into action, working with farmers, growing them, often experimental crops. Could you just share a little bit about that training program?
Very good. Thank you very much. So this program was part of that Gates Foundation as well as Ministry of Agriculture and Government of India. So what we were doing, that we were organizing these 2 weeks long training courses. We were bringing the breeders from the different countries. And in these training courses, we were not training them how to generate the experimental data set. Rather, once we get the data set, how to make the best use of this data set to create the next set of the varieties, working with the farmers, providing the better agronomy packages, because genetics and agronomy, they go.
Did they have enough science background when you?
Yeah, so they were basically working as a breeder, but in the traditional breeding approaches in the different countries. Yeah.
Thank you. I think almost every research program should be including that community.
Yeah, I think so. Yeah. And this has made the impact on that aspect. Yeah.
Yes, question, Francis.
So that was amazing. I just cannot believe that there's over 10,000 types of chickpeas. But this is a wonderful example.
Your butter chicken will never be the same again.
No, it's a wonderful example of international collaboration, but it must have taken so much logistics to get access to all of those.
Yeah.
Could you just expand a little bit on how you went about it?
Thank you. Thank you very much. Good question. So in agriculture, what happens for each crop, we are having these germplasm collections. They're stored in the different gene banks around the world. So what is happening that in ICRISAT, where I was working, this is a global research institute, even in chickpea, we had around 20,000 accessions, 20,000 different kind of chickpea varieties coming from 65 different countries. And these germplasm, they were collected before 1993. So at United Nations, there is a convention, CBD, Convention of Biological Diversity. So any germplasm collected before that, this becomes that germplasm held in trust, and now this can be contributed to anybody. When I was at ICRISAT, I had a plan to sequence the entire germplasm collections, but then I moved here. But good news is we already completed the sequencing of around 13,000 chickpeas. 7,000 left, somebody else will do that one. Thank you.
And as Foreign Secretary for the Academy, you would be very heartened to hear that response, I'm sure. Thank you. James, a question.
Thanks, Rajiv. And yeah, also, I didn't know that much about chickpeas either, so fascinating. I suppose extending that question, how's the current geopolitical environment impacting your research, and what threats do you see emerging?
So I think that the geopolitical issues, they pose challenge in two ways. One is this exchange of the germplasm from one country to other country. Second is even in terms of the funding. So even many of these, my earlier collaboration, we used to have a lot of funding from that internationally. But in the current scenario, number of regions, nowadays the funding from one country does not move to the other country. So what people are doing, they are working in their own countries. And then we are working through the data analysis and collaboration like that. Yeah.
Gosh, it's an extra challenge.
Yeah.
Thank you so much. Join me in
thanking you very much.
Thank you
very much. Thank you.